Okay guys, today we're going to talk about the female reproductive system. We're going to talk about oogenesis, which is the formation of the female gametes, the ova, and we'll look at a little bit about fertilization at the end of that process. So we're going to start with the oviduct, and at the end of the oviduct are these little weird structures called fimbriae. So this is the oviduct, and there's one either side of the uterus. So we'll go for the left oviduct here. We're going to have our ovary at the end of the oviduct, and these are called fimbriae. So when the secondary oocyte is released at ovulation, it's collected by the fimbriae and it allows it to move down the oviduct. Now the lining of the oviduct is a specialized type of epithelium. So epithelium is a lining tissue, and these columnar epithelial cells have cilia, and the cilia, the first job of the cilia is to waft the secondary oocyte that's been released at ovulation down the oviduct towards the uterus. So this would be the uterus. So inside here, we're going to get a secondary oocyte that gets released at ovulation at about day 14 of the 28-day menstrual cycle. So we're going to start with, how is the secondary oocyte formed? Well, the lining of the ovary is made from specialized germline cells, and they're called oogonia. So we're going to start with a single diploid oogonium. So this is now a cell, and it's a diploid cell with two sets of chromosomes. This is a cell in the lining of this ovary, so we kind of zoomed out a bit to get this picture. And that's going to divide through cell division involving mitosis, and that's going to form a primary oocyte. Now this is a diploid cell, and this cell is going to undergo meiosis 1. So you want to think primary and first meiotic division. So it's got two sets of chromosomes. It's the primary oocyte, but only one of these is going to further develop each menstrual cycle at the start of the menstrual cycle. So the primary oocyte then undergoes meiosis 1. However, this is the sort of interesting bit, that inside the ovaries of the female before she's born, all the primary oocytes are already made, maybe 100,000 per ovary, and they will actually undergo meiosis 1, but then they arrest actually in the first part of meiosis 1, which is prophase 1. So this is going to be an arrest. That means it stops at this stage. So all the primary oocytes will have formed their bivalents. So there's going to be 23 pairs of bivalents inside the cell, and then they stop at this phase, or they arrest at this phase, and they all remain like that until the female gets to puberty and she goes through the first menstrual cycle. So if there's 23 bivalents, we know the bivalents would look something like this. So that'd be one replicated chromosome, and then you have, through synapsis, the homologous replicated chromosome. This would be one bivalent, but we've got 23 pairs of them within this cell. Now, with the start of a menstrual cycle, this primary oocyte is going to finish meiosis 1. So what we're going to get is two daughter cells at the end of meiosis 1. Put that in there. Now this cell here is called a secondary oocyte, and this is haploid. So that means that the chromosomes have separated during anaphase 1, and then you had telophase 1, and then cytokinesis. So there's going to be one set of chromosomes inside the secondary oocyte. So you'll have 23 replicated chromosomes here, not pairs, because the pairs separated during meiosis 1. And the second cell is actually called a polar body. So this is not going to be needed, but it is important though. So the polar body doesn't really have any more function after this point, but it did allow the chromosome number to be halved from diploid to haploid, to go from the primary oocyte to the secondary oocyte. So this cell is really important, and what happens is that it undergoes pretty much straight away meiosis 2. So in meiosis 2, obviously the chromosomes are super condensed and we can see them, they're still replicated. There's 23 chromosomes here, and prophase 1 is going to finish. Sorry, prophase 2 is going to finish. Prophase 2. Metaphase 2 is then going to start. And in metaphase 2, they're going to replicate chromosomes are going to align down the equator of the cell. So this would be metaphase 2 here. And it arrests in metaphase 2. So much like the primary oocyte arrested in prophase 1, okay, we get development of the secondary oocyte. It's going to arrest in metaphase 2. So this phase is actually really, really super important because the secondary oocyte arrests in this phase. What we then get at ovulation, so this will be about day 14, there's a spike in luteinizing hormone from the anterior pituitary gland, and that stimulates the secondary oocyte to be released from the ovary, actually from the Graafian follicle inside the ovary. And this secondary oocyte, if I just draw this in, okay, it's arrested in metaphase 2, so it'll be something like this with 23 chromosomes, but it's actually got on the outside what we call the zona pellucida. So this is the jelly layer made of glycoprotein, this layer of the secondary oocyte. So I'm going to put here zona pellucida. Okay, that layer. So this is the cytoplasm and these are the chromosomes in metaphase 2. But also on the outside of the secondary oocyte, as it's ovulated, it's got some of the follicle cells from the theca of the Graafian follicle. So as the Graafian follicle ruptures to allow, just do these like this, release of the secondary oocyte into the oviduct, some of the follicle cells remain outside, and these cells are called the corona radiata. So that's the structure that would get released at ovulation, which is at day 14 of the menstrual cycle. Now, what happens is if a sperm cell manages to fertilize this secondary oocyte, you've got the acrosome reaction as the sperm cell gets through the corona radiata and makes contact with the zona pellucida. So the sperm cell would have exocytosis of hydrolytic enzymes. They would hydrolyze the protein in the zona pellucida. So they hydrolyze peptide bonds to digest the pathway through it. And then the sperm cell injects its nucleus into the cytoplasm of the secondary oocyte. And that stimulates meiosis 2 to then finish. So what would happen here inside the secondary oocyte, so I'll try and do it up here, is you get this scenario. So we'd have the sperm nucleus inside one of the two cells, which is now called the ovum, because this cell here, the secondary oocyte, has now finished meiosis 2. So here you'd have the nucleus, okay, with these two sister chromatids in. This one here would have a nucleus as well, but this is called the polar body. So the polar body actually doesn't have any role to play anymore. That's not actually useful. And the sperm nucleus is in the cytoplasm, and this is now called an ootid or an ovum. So what we're going to get is this nucleus here is going to fuse with this nucleus. So you get a bigger nucleus that's diploid with two sets of chromosomes. So this would be the ovum nucleus with two chromatids or two chromosomes. For example, in our picture, this would be the sperm nucleus with a haploid set of chromosomes, so two chromosomes in this case, and they fuse together inside the ovum cytoplasm. So this would be a fertilized egg. And again, on the outside of this, the zona pellucida by this point has actually become what's called a thicker, harder fertilization membrane. So as soon as the sperm enters the secondary oocyte, it causes cortical granules to release chemicals by exocytosis, and those granules cause the zona pellucida to thicken and harden to become the fertilization membrane. Now that's important, the fertilization membrane, because that prevents a process called polyspermy. It prevents other sperm cells injecting their nucleus into the fertilized egg. Now this ovum is then going to divide through repeated mitosis. So I'm going to have to do this a little bit smaller in the gap here. And because it's got lots of cytoplasm and lots of organelles inside the cytoplasm, it doesn't need to get bigger or rounder, anything like that. We get replication of the DNA from the diploid nucleus after fertilization, and then we get mitosis. So you'll get cytokinesis right down the middle. So you'll end up with two cells, both diploid, obviously separated into the two separate cells, and then they keep dividing through cell division with mitosis. So what you end up with is a big ball of cells, which is the embryo. So I'll just do these in our picture here. So imagine these were like individual cells. That's the ball of the embryo. It's called a blastocyst. Now the blastocyst, if you go back to the ciliated columnar epithelium, what happens is their second job is to allow the blastocyst after fertilization to move down the oviduct in that respect. And the wall of the uterus is a thick endometrium. So I'm just going to draw this in. So that thick endometrium, let me just put endometrium. That allows implantation of the blastocyst, where the blastocyst will further develop, and it will develop the trophoblast villi, which allows a big surface area to absorb glucose and amino acids, within the endometrium. And it also produces a hormone called human chorionic gonadotrophin. So the chorion on the outside of the blastocyst eventually produces HCG, and what HCG does is it keeps the inside the ovary, the corpus luteum, making progesterone. And that progesterone that comes from the ovary, from inside here, the corpus luteum, will actually maintain, so this is progesterone, the thickness of the endometrium, but this progesterone, from the corpus luteum, which is maintained in the first 12 weeks of pregnancy by HCG from the blastocyst on here, is also going to inhibit any further production of follicle stimulating hormone from the anterior pituitary gland. And that means that, guess what, there's no more primary oocytes going to develop. So this process here at the top actually requires follicle stimulating hormone, which comes from the anterior pituitary gland. And that means that as the pregnancy proceeds, there's no more periods, and there's no more primary oocytes that would develop inside the ovary.